in my talk and start, with a broad view on why should you be the next generation, why it matters that you are entering in plant science and all your work, why it's important that you are doing that. And then I'm speaking about next generation breeding. I'd like to give also some context of what I believe is next generation breeding, why it's important to do it now, what are the problems we are trying to solve. And in this context, why having resources like the wheat genome sequence and the quality that we have achieved matters, because that has been really an important fight actually to get to this sequence and to the quality of it. So what we have achieved, and beyond what we have achieved, what did we learn? Because it has been also a great adventure to do all of that, and I have learned way beyond the scientific things in this wheat genome sequencing journey. And as Alexandre mentioned, I'm going to give you a flavor of what I'm doing now since 1 year with INARI in Boston. So we have a very highly complex challenge to solve all together, and everybody has a place and a part to play into it. It's a very relevant one, and you have all heard in many talks about we need to feed an increasing amount of population. But I would say if we only stick to that, we are probably answering an incomplete question, because we are at a stage at the moment where it's not only about feeding people, but it's also about protecting the environment. It's also about doing that in the context of climate change. We had great talks, illustrating what we are facing. We have to do that also by taking into account there is more and more demand by the society to have, healthy food. And we also have to do that with farmers that make a living, because farmers are not here to make a social work. They have to have a living too. And their situation and being a farmer is very tough these days. So this is a very complex equation to address actually, and I just reformulate it. Is for us as breeders, it would be how do we quickly develop new varieties that are health promoting, that are performing locally in their environment the best as possible with the least input as possible, which is not the way breeding and agriculture production has been done in the past decades, and while we are enabling farmers to make a living as well. So plant breeding, for those of you that are not very familiar with it, has a long history. It started 10,000 years ago. We had our Neolithic ancestors that were starting to select without knowing what they were selecting for. I mean, they knew what they were selecting for, but not what was behind, because before the 19th century and Mendel working on the law of genetics, nobody had an idea of heredity and how things were working. We also had, breakthrough when Rosalind Franklin, Watson, and Crick discovered that behind this whole genetic information there is DNA. Then first really breakthrough in breeding was the Green Revolution with someone called Norman Borlaug. I hope you know this name. He's one of the few scientists that won the Nobel Prize for Peace, given the impact it had. So also a very inspiring person, and he worked on wheat, so even more inspiring. Then we had, in the 80s, the early generation of GM and BIOTECH, booming up. And then since the 2000s, we're in the era of markers and, genomic selection and using, the information of the genotypes to do a better selection. So this has been quite successful because, we have increased the production. And so if you look at different statistics, you can go on many websites to look at. So I have wheat in the middle. The wheat increase, you see, since the 60s, there have been really a strong increase. You look at average corn yield, same. All over the world, generally the yield has increased. So it has worked. But it doesn't work that great anymore. So you look at soybean, for example, on the left side here. Well, generally the soybean production is increasing in South America in particular. But the yield itself is not increasing that much. What is increasing is the expansion in the land. And this is at the expense of the Amazonian forest. And that is not a good trend. So there is a problem here of the capacity to increase yield per se. You look at wheat, that's even more damaging. And I cry when I see that, because it's really hard for wheat to cope with the fluctuation. And wheat is very susceptible to climate change, actually. And so we are even having yield decrease now. So that is since the 60s. We had an increase since the 90s. This is stagnating or decreasing. And what is perturbating as well is for the farmers. You look at the corn here. So this is also the yield increase. So it's steady, it's increasing, okay. But the price that the farmers have to pay for corn has nothing to do with the genetic gain they get for the price they pay. And that is also not such a great situation and not a sustainable way of doing things. Now, when you look at traditional breeding, I mean, you make crosses, you select, the outcome of the crosses. I mean, it takes a long time because you have to do thousands of crosses and you do not know really what you are mixing together. You take the best of the best. It takes a lot of space. It takes a lot of money to do that. So if you look in the industry, there is more than $5 billion spent per year in breeding. In general, for the main crops, it takes 10 years between the first crop and what you can bring to the market, or 15 years, so it's really long. And then you do all of that for having at max a 1% increase in genetic gain. So in my eyes, this is pretty inefficient. And when you see everything, evolving around and you think we still have processes that take that much time, energy, and money, I mean, there is something that we should do about. And one of the other issues is that by focusing so much on yield, and we had already bottlenecks of domestication, then there are now bottlenecks due to selection. I mean, the diversity in the pool of elite varieties has been reduced, and diversity is the essence of breeding, and so we have a problem now of how we bring back diversity into the pool. And there are a lot of gene banks, but they're actually not well used because it's extremely difficult to, no breeder wants to make a cross with a wild relative. I mean, you can ask to any breeders, it's horrible, it messes up everything. So that is a problem. So we have things sitting in gene banks and we cannot use them properly. Then if you look at the biotech, so the GM traits, I mean, that had some success. It brought, herbicide tolerance, insect control, so it has helped to increase the yield and protect the yield, but that's the same. And since the first years of 2000, there is basically no progress. And plus the GM technology, has some advantage, but this is also not a very subtle technology. You overexpress genes. You can't put a lot of genes at the same time in a genome. You have a lot of silencing. So it is also not, in my opinion, the technology of the future. And when you look everywhere in the R&D industry, this is actually, the pipeline is pretty dry on the GM. And it is not a technology that can deal very well with very complex traits like drought or yield. I mentioned the impact on the environment. This is way too high. I mean, 70% of the water in the world is used in agriculture. Nitrogen, phosphate increase. So there have been a lot of efforts, for people to reduce the use actually of input. But for developing countries that also have the right to develop their agriculture to feed their people, where you look at the increase of nitrogen and phosphate utilization in China, for example, that is also not such a sustainable solution. So the way the varieties are bred is pushing for that. And the result of that is ending up in the waters and the pollution that we hear about in the atmosphere as well with the green gas emission. So, and finally, something that I think will be also a problem of your generation. It has started with mine. We have not been doing a very great job at tackling it, actually, so I'm counting on you guys to do a better job. It's the disconnection that the society has with science and not understanding the benefits of the technology. Everything is a risk, nothing has a benefit anymore. So we moved in agriculture from something that people thought, okay, it's the beginning of civilization is agriculture, we are where we are because of agriculture. Farmers are great. They are feeding people, to farmers are polluting the environment, and we don't understand what is in for us. And then we are again, and since I'm French, we are very good at leading the pack in the world. Unfortunately, not the rugby one, but this one. So that is really something important because I always mention to my teams is that we can have the best technology in the world. It can save the world. If people don't want it, you are done. So that is something that I think, and hopefully I hope at the universities are more and more is communicating about why we do things and how we do it is very important. So there is no panic to have because I think there is still a solution. So we should keep calm and start a new revolution. It cannot be the same kind of revolution like the one that was driven by Norman, but it has to be in the same spirit. We are here to help and have an impact and save lives, and that is working in agriculture and in plant research is a very purpose-driven mission, I find. So that is great to be here and for you to do that. So how we do that? So the vision I and other people have is that okay, we have to do breeding in another way. It has to be better, it has to be cheaper, it has to be faster, and it has to remain accessible to all. Something that pains me is how the biotech and the GM technology became something that was only in the hand of the big companies. And they didn't fight it, because the higher were the regulation level and the entry barrier, like we call it in industry, the better it was for them, because they were the only ones who could afford to pay 150 million per deregulation per GM event. It should not happen to this technology. So there are a lot of debates, and I'm going to cover that in one slide. So what we have to do, and I find this really exciting, is that we were in a world of reading only. We were reading the phenotype, we were reading the genotype, and that's all what we could do. But now, thanks to the revolution that is happening in genome editing, we are suddenly entering an era of read and write. And that is fundamentally changing everything, and that's why I'm so excited about the fact that we can really, I think for the first time, completely transform breeding. So by being able to edit genomes, we will speed up the breeding cycles, and we will be able to create new diversity and addressing the issue I have mentioned. We also, because of all the capacities we have, and we have some illustration in this conference and others, we can move from simple and trying to simplify life to the reality of biology. The biology is complex. We have seen some of it. The talk of Stella, I think, has illustrated it as well. It's not easy, to do things. There are interactions everywhere, but I think we have resources now to do it. The whole capacity to computationally analyze data. Data are not very expensive to produce anymore. You can, use machine learning. You can do a lot of things that even 10 years ago nobody was dreaming about. So I think we are here able to suddenly address more complex traits and in crops and in more complex genomes than just working on model system. And second, we have to move also from breeding for varieties that will fit more or less everywhere, because it's so expensive and long to produce a variety that you have to sell it on as many hectares as possible, to having things that are very adapted to their environment and will perform the best genetic for the best environment. So being able to predict in advance what is performing in this environment, and if you have a development time and a cost of developing a new variety that is way cheaper than now, you will be able to do that. At the moment, there is no incentive to do that because it's too expensive to develop a variety. And we have to take into account, that a plant is living in its environment, microbial, all the environment, and so all of this interaction matters. So there is complex biology to do behind that, but again, I think we are at the time where we can start to tackle that. And the acceptance of new technologies I have mentioned, that's also something important. So the ingredients of breeding by editing, which is for me breeding 2.0, is first of all discovering and designing. And that is changing from the breeding mindset that we have for the moment. It's like you do things randomly and you cross, and then you basically select at the end of a long process. Here, because you can edit, you should be able to discover the genes you want to edit and design the kind of crop of variety you want to edit. And then you, of course, need an editing toolbox and the capacity to deliver your changes in the genome in an efficient way. So all of that has to come together to make breeding 2.0 a reality. And then you also have a regulatory environment that needs to be here. So in the first, and something I will touch upon in one slide is also nobody will be able to do that alone. I have been involved in different public-private partnerships. I think it's more than ever very important because you can learn from each other. I've been in the two worlds and I really see a complementarity with both. So don't be afraid about the private guys, and and then choose wisely with whom you want to work. You have to also be requesting things. But there is a lot of drive and a lot of efficiency that you can benefit from. Okay, so discovering and designing. So the simple question is, what do I want to edit and how should I edit it? What kind of modifications should I do? And that has to be driven by the biological question, the trait, the whatever you try to improve. And so how do you identify a relationship between your genome, your genes, whatever, your sequence, and the phenotype you want to have at the end in your field? And this is where I will spend some time explaining why genome sequences are very important, and having one genome sequence is not enough. So the capacity to have high quality genome sequence that are well annotated, that you know exactly what genes, what they are doing, what the hypothesis behind. The capacity to resequence all of your diversity panels in your crop and having population to do association genetics, having mutant collection. You see it in all the talks. All of that is really important, and having a genome sequence is just empowering that. And then you need to produce data as well to link to the phenotype. So of course, data in the field, it becomes easier to produce data in the field. You have a lot of drones and new technologies now that are really bringing new capacities to do phenotyping. And I think as well we will need to also have a better system to basically predict the performance of the plant in the field, even at the cell level. Because if we can have the attrition at the very early stage of a screening and then go to the field to check, that would be the ideal. I probably will be retired or more if when this happens, but we need to work on that. And we cannot do that without having all the capacity in computing and machine learning, because I don't think we have enough in our brain to manage all of that. So going to the genome sequence and illustrating the adventure of the wheat genome sequence. So in 2000, and when I give these talks, I realized you were probably at school for most of you in 2000. So you have not realized how breakthrough it has been that we got the first genome sequence of a plant. So a really good quality sequence. I mean, that was really great. But at the time, there was the idea that that's going to be enough. We will have that and we will answer all the biological questions. And there are even some people that have said it would be totally stupid to sequence corn and to sequence wheat. So you have also to be careful what you say when you are asked by journalists, because it sticks in the journals. So actually now, 18 years later, we have more than 300 genomes that have been sequenced. You see there are an explosion. So this has only been possible because the cost of sequencing has totally dropped, thanks to the Human Genome Project that has pushed the technologies. But that's great, because you don't want to spend, like me, your career sequencing genomes. You just want to do that in 2 days. And when I did the year before the PhD, it took me a year to sequence 16 kilobase. Yeah, by hand, and you read the things, and when you read the 200 base pair, your day is done. Yeah, and I'm not 80. So it's very important to have foundational genomes to really understand the biology of the crops. And here, this is something I drew, in 2008, I think, when we were preparing a review. And this is the number of QTL cloned in rice after the release of the genome sequence, and at the same time, the number of QTL cloned in wheat. No genome sequence. So that was really motivating us to say, okay, we need to increase things. There are different kinds of quality of genomes. Usually people always say they have a reference sequence. Well, there are different qualities of genome sequence. There are draft genome sequence that are basically not completely assembled, aligned along the chromosomes, and that's good enough to do a number of studies. I mean, comparative genomics is actually getting really interesting results with this kind of genome. So you don't have to get always the quality for everything. But when you want really to enter into the biology of traits, you really need high quality, because you need to get to the genes, the regulatory element of the genes, and understand how all these things work, and then link that with the transcriptomic, with all the omics of the world. And that was the vision of the Wheat Genome Sequencing Consortium. So we had this vision in 2005, and we were not a lot in the room. So we were at the Plant and Animal Genome meeting. We were about 10 in the room. I was there. There were people from the Kansas Wheat and the Kansas State University that were really actually pushing, because they were jealous of what is happening in corn, and they say, we need something like that, I think, because we need to accelerate the improvement in wheat. So they hired an executive director. And then Limagran was one of the companies that also said, okay, we are going to help you doing that. But we were here and saying, okay, wheat is a 16 gigabase genome, 5 times bigger than the human genome. It's polyploid. You have 3 genomes to deal with. And so we had the debate between the scientist and the breeder, because the scientist said, I mean, hexaploid, really? Why don't we do a diploid, an ancestral relative? But then the breeder said, yeah, I mean, do you want something that is useful for us? If it's that, you have to sequence the hexaploid genome, because 90% of what grows in the field is hexaploid. So you have to know what you want. So we had interesting debates for a few years. And then how do we sequence that? At the time, sequencing it whole genome or doing a back-by-back, and everybody said, back, this is such a has-been approach, but some of us said, yeah, but I mean, to have quality, that's the only approach at the moment. So we devised a strategy, and we basically went for it and said, okay, we want a genome that is useful for the breeders. If you want to be with us, come with us. And so we want to empower gene discovery, gene improvement, marker-assisted breeding, and so on and so forth. And therefore, we did the deadly decision to sequence the hexaploid with genome and have a high-quality reference, and and have put a roadmap together to reach that. And at that time, many people thought we are just nuts and we will never make it. But that's usually where I get motivated, when people tell me something like that. So I drew this cartoon, I think, in 2009. And I looked at it last time and I said, I had the time quite right, actually. Like the high-quality reference genome sequence was published last year, somewhere in 2018. And we have changed a bit the way we planned to do it at the time, but actually the timing was right. And then the vision of what we would provide along the way, having different versions of the sequence until we get the high-quality sequence. And a few of the the cultural parts of the commitment we had of working together is that we will constantly evaluate new technologies to do things, but we will never compromise on the quality. We will never tick boxes, and we will never just do it because we can have a publication in Nature faster. And that was really important to have that, and actually binding us together to do that, because nobody could do it alone. So it was really also a human adventure in that matter. We organized also regular workshops, and there was a lot of collaboration, because at the beginning we just distributed chromosomes by countries. So there the executive director had a really brilliant idea, because we had difficulties. So we traveled around the world together for a year trying to convince the funding agencies, can you sponsor a chromosome with your country and things like that. And then it didn't work very well. And then she said, because I had a project in France with the ANR to sequence the first chromosome 3B, 1 gigabase. And then I had the back library, so we started already a bit in advance. And then she said, let's put the French flag on the map. So we put the French flag on the map with chromosome 3B, and then you cannot imagine, everybody said, we are not going to let the French take the lead. And suddenly, everybody in the US, in the UK, say, okay, I take a chromosome, and suddenly, 3 months later, we had about half of the chromosome sponsored. So that was one of the brilliant tips I can give you for international consortia. It was also important for us that we support small companies that didn't have their own research, and so that whatever we were producing was useful for them as well, even if they didn't have a big R&D department. And then providing early access to users too, so people had access to the product, the sequences way before publication, that people that were in the consortium, and in particular the breeding companies. So I mentioned we were 10 in the room in 2005. Now there are more than 2,400 members, 68 countries are involved. So it is a big success. I think it's one of the consortium that continues to hold on and is now in the second phase. I have let them go in the second phase because I have other things to do. But this is something I'm very proud of, having been part of this adventure. So a bit science. So how did we do that? So I mentioned we started with BACs, and we did actually physical maps. So first physical map was done by the group I was leading at INRA, chromosome 3B, published in 2014. Then we sequenced it with the Genoscope, and then 8 chromosomes were done this way, and now there are more. Then we also did in 2014 chromosome sequencing, because we have a great colleague in the Czech Republic who is able to sort chromosomes. So we have specific lines where you can isolate each of the chromosomes independently, and when Illumina started to boom, we basically sequenced every of the chromosome by Illumina. So it's definitely not a reference sequence, but we could assemble per chromosome the gene set, which was already an advance compared to what we had before. And then the really breakthrough was with NRGene, when NRGene came with an assembler that was able to assemble the whole genome of wheat. And assembling the whole genome of wheat, there are 86% of repetitive sequences. It's like if you have a puzzle and you have about 80% of the pieces that look the same, or almost the same. So it's complicated to do whole genome assembly, and the algorithm were not able to resolve that until NRGene came with a really cool algorithm, and the quality they had, so we checked and we were say, okay, show us really how it is. And actually they had a sequence 3B on their whole genome assembly that was as good as the one that we produced at INRA by BAC. So what we did with the consortium is that we mixed all of that together to have the best sequence we can. We added bio-nano maps and IC analysis to really have an order, and that was what was published last year having the first reference, and I'm happy to announce that next week they will actually release a second version of the genome where there have been additional bio-nano map done on the whole genome by the colleagues in Jan Vorak's lab that will continue to increase the quality. So in few numbers, what was exciting for us, and it may be not exciting for you if you are not in genome sequencing, but this is exciting because the N50 is the size of the contig in an assembly that is representing half of the whole genome assembly. And until the whole genome assembly and our back libraries, I mean, the N50 was about 100 KB, 150 KB, 1 megabase when you push the technology, and suddenly 7 megabase. So it means we had large contigs, and large contigs mean well-assembled genes and information that you can really annotate properly, and you can put that along genetic maps and order your information and have pseudomolecules. So it was really great to be able to assemble this 21 pseudomolecule. Beyond this result, what for me is important is the impact, because you can publish in Science and it's great, but if nobody is reading your paper or if nobody is using your sequence, in my opinion, you have not really reached your goal. And so we were looking at how many people are using the sequence. So we have a website where you can download the sequence and you can blast, and there have been more than 1.2 million blasts on this sequence, and that makes me happy. And the number of downloads as well of the sequence, so it means people are using it, so it must be somewhere a good product that people like it. And I think there will be some illustration in the next talks about what people are doing with the sequence, so it's great. And what made me happy too is to see we were looking also, looking at all the papers that were published in the past 2 years. So first of all, there is like the curve I have shown for rice, now it's happening to wheat. Cloning a gene is taking 2-3 years maybe, and when I did my postdoc, it took me 10 years to clone one disease-resistant gene. That's where I got mad. That's why I'm like I am now. So I said I don't wish that for the next generation. Therefore, I will sequence the wheat genome. But what is really interesting is to see there is an explosion in the use of the genome for breeding and also for trait genomics. So I think for me that is really satisfying to see that happening. Okay, so now expanding again, we have genome sequences for many crops. Resequencing is happening all over the world. So what do we do with this and how impactful it is? Well, this is the essential information when you combine it with other omics information, with the phenotype, and we need more of that, and we need more of that coordinated. So that's also a call, like, many people are doing their experiments in a certain way, in certain conditions, and actually there is a lot of data, but since there is not a lot of coordination on the experimental design, we are losing a bit the momentum of the capacity to produce a lot of data. So I would call for more networking and more organization of producing data together and having ways of experimental designing things together. So having a sequence, you can look at genes, do genetic analysis, identify the genes, and beyond that the gene networks, because biology is more about gene networks than individual genes. And then you can also, actually I don't care about the genes, but I can identify, do a quantitative genetics approach and identify sequences that are positive or sequences that are deleterious, and have that as a target as well for breeding. And that, for me, is the source of what I want to do in the second phase, is editing genomes based on this information. So if you have that, you suddenly are in a position to understand what you want to edit, and then how you want to edit it, and I will come to that in a minute. And if we have that, if this is done properly, we will be able to engineer complex traits and boost genetic gains more than what we can do at the moment. And I have 2 slides to illustrate that. So there have been a lot of success, in disease resistance. You have one gene, one trait, so that has worked very well. Many genes are cloned. We start to understand, listen at the talk this morning of Tela, we get more and more understanding of how things work. Then you have an example here of old composition. You have a few genes responsible for the characters. There have been progress here as well in deciphering that. But in yield traits or drought or anything that is more quantitative, and when you look at human genetics, like our height, is governed by hundreds of genes. So when you look at papers in human genetics, like 800 genes explain only 16% of the variance in human height, whatever here. So it means there are hundreds of genes. So I mean, how do you do a breeding, not in human, but in plants, how do you breed for that, you know, when you have no clue about what you are breeding for? So you need this knowledge, and having this knowledge will enable to really crack those. And the second one is something I borrowed from John Hickey's lab. So John Hickey is at the Roslin Institute in Scotland, and he has done really great work in genomic selection in plants at the beginning of his career, and now in animals, and actually he's combining both now. And he has now, you know, he's promoting an idea of called PAGE, like promotion of alleles by genome editing. And basically what it shows here is that that would be the curve you get of, you know, over generation of the genetic gain you have by genomic selection. But then you add genome editing on the top, you know, 5 edits, 1 edit, 100 edits, and then you say if you can do 100, that's the best. You know, you get suddenly, instead of maybe a 1% increase or less than 1% per year, you get to 3–4%. That is not possible with classical breeding anymore to get this kind of increase. So combining, you know, this technology that we know with the new technologies has really a lot of potential, and that is something that is feasible in plant breeding, and that's something we're after at INARI. Okay, sorry, the second part of it is it's great to actually, let's imagine we have the knowledge and we know what we want to edit. Okay, how do we do that? So for those of you that are not familiar with the, I don't know how you would be because it's everywhere in the news all the time, CRISPR, you know, there are enzymes that are able to cut the DNA at a very precise location. They are, in the case of the Cas nuclease, they are guided by a guide RNA. And so there are different kinds of modification you can do with that. So if you bring the nuclease at a certain point, you let it cut, then there are repair mechanisms that are actually a bit sloppy and create deletions or small insertions at the site of the cut. You don't do anything else. But you can also provide a template for repair, and this template can be inserted or can help you to correct or add new nucleotides in a certain piece of your DNA. Then people have engineered even further this kind of system, so you can use the Cas actually and a modified Cas that doesn't cut anymore, but that is fused with another enzyme that has another property, and this is used here. You have a Cas fused with a deaminase, and what it does is that it's changing nucleotides. So there are deaminases that change a A into a T. And so that is also based, because you want to be precise, on the Cas and the guide bringing it to the place where you want to do the change. And you also have now systems that have been developed to also put a Cas that is inactivated with a complex system that I don't want to explain here, but that are bringing methylase or demethylase. So you can also methylate and demethylate, you know, the DNA at the place you want. And we know that methylation and demethylation are acting on the expression of promoters and the expression of genes. So you start to have here an idea of, you know, the diversity of changes that you can do, and the one that work the best actually are in this category at the moment, because as usual they are the simplest, so we rush for it. And so there are many projects on the way to do that, and you have some in your lab, and I've seen posters, and that is great. And when you look at the commercial pipeline and what is cooking as well there, well, there are some startups that have, you know, made their business on that. And so with single gene and loss of function, there are examples in soybean that to modify the oil quality, actually in potato for the cooking quality as well. So this is on the way, this is in the pipeline of industry, and it works well. There are also derivative of the first technologies developed here in the lab of Zach Lipman at Cold Spring Harbor with one of our advisors. And what Zach has done is that he's directing 8 guides to a promoter of a specific genes, and by doing that he's creating deletions a little bit everywhere, and a population of tomato in this case that has a lot of new variation, because you have basically, you create mutants in your promoters, and then suddenly he has a range of variation that he had never seen before, even in classical mutagenesis. So you increase, and it's not changing the coding sequence, it's changing the expression, and it creates new weak alleles, and that is really useful for breeding, and we have a license at INARI to use that in a commercial pipeline. But this will not be enough. We cannot do the whole biology by knocking down a few genes. I mean, the biology, as I mentioned, is very complex, so we need to multiplex. So that is a big theme also, is we need to have the editing at the level of breeding. And when you do breeding, you have to modify hundreds of genes at the same time. So this is taken from a paper of George Church. I don't know if you know George Church. He's in Cambridge also. in Massachusetts, and he is leading a lot of human genetics, and he's pushing the boundaries of research in genome editing. And from this paper, you just have to look at, that's where we are at the moment. If you want to actually use the Cas system to do a lot of cuts, you will have a problem because the cell is actually dying. It's reactive negatively to too much cut in the DNA, which hopefully it's good for us. Huh? But so you can't edit 50 genes with the current technology. So we will have to find ways that either you have less cuts and you still can do your modifications, or you have bigger pieces of DNA you can introduce to do your modification. But that's definitely something that needs to be worked on. There are a few examples of, reaching high copy, high modification of genes. But in fact, when you look in the details, so in sugarcane, there was 107 edits done, but that was one target because the gene was in 107 copies. So it was one guide and one gene 107 times. So there are examples that show actually, the cell can tolerate some things. And so this is new area of research, but very important, in my opinion, to deploy the breeding 2.0. I mentioned, the difficulty that we had with the GM and the regulatory, and you may not be familiar with it because it's something I learned when I joined Bayer. Actually, when I joined Bayer, I learned a lot of these things, regulatory, stewardship. And this is important because, we know GM is regulated, and regulated means you have to do a lot of studies because people in the public think you do a GM, you throw it in the street and nothing is done. That is not true. Huh? There are 3-5 years of testing, of safety and all of that. That costs a lot of money and that takes time. And the more you ask for that, the more you penalize the small companies that cannot afford it. So highly regulated environment is not good for innovation and for the small companies. In breeding, and that's the irony of all, in breeding you mess up with the genome, like you cross two things, everything is messed up, you do irradiation also to create mutants, no problem, no regulation. And in the middle, you have genome editing with a big question at the moment in different countries. How are we going to consider that? Is it a GM? Is it like breeding? Is it in the middle, depending on the modification you do? Some modifications are closer to breeding. Some modifications are closer to GM. So there is a lot of debate at the moment. And to me, that's also something that you need to also integrate is these kind of things, because as I said, you can have the best research in the world. If you care about the impact that your research has as in is applied somewhere in industry, well, you have to have a look on these things as well. And we are in Europe here, where we look at the situation at INARI of how the countries are starting to speak about the regulation for genome editing. So green is good, red is bad. Guess where it's bad? Okay, last year, you have maybe heard it in the news, there was a ruling by the European Court of Justice to actually say, actually everything is GM, but we are going to exempt a few things. So we exempt classical breeding, we exempt mutagenesis, but we don't touch at the moment. Genome editing stays in the bucket of GM. I am not completely hopeless because I think people have realized that this may be a mistake to do that again and then hamper the capacity of small companies and the potential that it has for breeding. So a lot of people in the public sector actually are fighting, and that's good. I think the company should stand back, don't do anything, because that is not good when companies start to lobby on these things. But in the US, in South America, in Australia, even in Japan, so countries that were traditionally also against GM are actually now considering that genome editing is okay. So I see that as a progress. And it's important that we continue to have this debate and explain, what are the potential utilization. And we should be very open about what we want to do, how we want to do it, to have an open debate about it. All right, public-private partnerships. So all along this project, and in particular with WIT, I had the possibility to put together public-private partnerships. I had a project in France called BreedWIT that is actually just finishing now. The IWGSC was a public-private partnership, and it was very interesting actually to learn how to work together. And so one of the things that I think is important is to define the objectives at the onset of the project. Having been in both worlds, often people in academia think that they should write their project, knock at the door of industry and say, can you fund my project? That is not how it's working, because often your project is not aligned very well with what industry. Sometimes it's not very far, but sometimes it's too far, and therefore the dialogue is more difficult. So there are more and more incentive in the funding agencies actually to bring people together at the beginning, which I think is great. And so I would encourage you to really discuss what you are after, and you can still do your work and what you are passionate about and publish if you do that. And that's the second point I want to mention. It's very important to understand what are the drivers of each other. In the academic world, it's important to publish. And industry has been long to recognize that, and often was sitting on the result and slowing down publications, and that didn't really improve the relationship. And on the other hand, industry was very nervous because scientists wanted to speak about everything, and then in terms of IP, that created problems. And so you have to understand that industry lives also by keeping things for a moment and putting IP on some of the aspects. So it's important to have that at the beginning. I would say it's as important as when you discuss about the authorship for a paper with collaborators. You should do it at the beginning to avoid, a disaster at the end when the paper is ready to publish. To make that easy, it's good to discuss what is competitive and pre-competitive. Industry has learned that, there are some part of the work, like the wheat genome sequence, people in industry never had the problem that this will be public. Never wanted to put IP on it, never wanted to patent the sequence, because they consider this is fundamental information, this is what we will do with it, that then we will patent, and the product that will derive from it. So it's good also to define, okay, is this something we can share together, and then competitors were together in the consortium working together on making the sequence and then competing again later on. So this is the kind of discussion that is also very important to have. And my own bias, I have realized when I arrived in industry, is that I was not completely sure what I will see there, because I never saw these guys in conferences, so I didn't know if the researchers are good actually, and they are really good. I was really, I have to admit it, I was really astonished how great the scientists were at Bayer. And there's something also we have to be careful not to be biased and think, well, okay, because they are industry, probably it's because they failed their academic career. That can be a choice to go to industry. So it's important to keep that, and then don't hesitate to ask them to lead a work package in your project. I mean, that is great, to learn from each other. Everybody has to learn from each other. All right, so 3 minutes to finish and to share with you my new challenge and where I have fun since 1 year, so at INARI. So I've tried to capture our mission globally. That comes back to my first slide, is we want to be able to contribute to fill the world without starving the planet. So that's a very short summary of what we do. And we want to do it in a different way of what has been done before. So making it happen, but the way we will make it happen matters as much. And we are an interesting mix of people that had been in big industry. We have people that worked at Monsanto, at Syngenta, at Bayer, and all the big ag, And then we have people that never worked in industry and come purely from academic research. But we know what has worked and we know what we don't like with the current system. And so that is a big driver of doing things in a different way. I want to spend a minute to explain you what is the thinking behind, and it is already what I have presented somehow. It starts here with a computational genetic design. So we want to be able to predict genetic pathways and network. And we want to basically be able to predict also ideal crosses. So we want to design at the early stage of the breeding process, not wait until the end of the breeding process that we have by chance what we want. So this is a lot of work, and this is still in the infancy because there is a lot we don't know about plant biology and how plants are regulated. But we start with that. Then we are building up a toolbox, and I mentioned all the different kind of edits. What is very important for me as leading the science organization there is not to be technology driven. I actually don't care what tool we are using as long as we can do what we need to be doing. So if we understand here, we have a network of 50 genes, we need to knock out 10, we need to increase the expression of 5, we need to decrease the expression of 15, and so on. I want to have a toolbox, whatever it is, that will enable me to do that. So I want to be driven by the biological question, not by the technology push. And that's another thing, I want to advocate for. It's easy these days to be driven by technology because technology is super attractive, it's easy, it's fun. But I would try to stick to the problem, the biological problem. And then the third part, and I didn't mention it there, but at the moment, and I think Stella mentioned it as well, transforming plants is still not very easy. It takes a lot of time to get things. And often, when you are in an industry pipeline and you want to have your modifications in the elite varieties that you will sell, you can usually not transform the line that is in a elite background. You have to transform something that is not really good and back cross and back cross and back cross. So it takes years actually to do that. So the dream would be to be able to deliver your edits in any varieties and immediately. So that would shorten the time by 5 years. You would have it in 1 year. So we are also working on new technologies to do also this kind of delivery. And then in our development, we are not going to sell seeds directly to the farmer. We are partnering with growers that know how to multiply seeds, how to do their whole business, and we are going to give them parental seeds for their breeding programs. And so we will be able to scale up, because it's important when you are in industry. I mean, you can't just sell a bag of 25 seeds to a farmer, so you have to have a whole logistic that is very important to have together. So with that, we have the vision to have improved seeds in 2-3 years rather than 10, and for a $5 million cost rather than 100 million. So that's quite a bold statement, but we are all super motivated to make that happen. And what is very important as well is that we will learn and we will have this iteration of this foundry cycle, and we will become better and better at doing that. So our first product will not be revolutionary, I'm sure of it, but hopefully the next generation that will come will really prove the point I'm trying to make here in this talk. So who are we at INARI? We were founded in 2016 by Flagship. Flagship is our investors that actually, they are located in Boston, and they create a lot of startups in pharma and now in agriculture. We have started in Cambridge. We have a research site that has started last year in Ghent. So we have 8 people in Ghent at the moment. And we have a production site in the Midwest, in West Lafayette in Indiana. Because if you want to be credible as a breeding company, and you tell farmers that you are doing breeding from Cambridge, Boston, Massachusetts, they are not completely buying it. If you tell them and you bring them to somewhere in the Midwest and they see your field trials, they are a bit more confident that you are serious. And this is the leadership team I'm belonging to. And another thing I want to point out is that there are 50% of women in this leadership team. If you look, I encourage you to look at the board of all the big companies. You will see that the gender diversity is a bit less fun. And so our CEO is a woman. She's from Thailand. Here is Julie Borlaug. So Borlaug should remind you the name because she's the granddaughter of Norman Borlaug. So she's quite well inspired because she swore to her grandfather that she was going to continue. So working with her, and she loves wheat too. So we are the 2 wheat defenders in the company. And so Mark came from Limagran and has worked with Monsanto early in his career. These 2 guys come from Syngenta. So he was in a bank before, and he loves the story at INARI. So all of us are here because we really want to make a difference, and we have fun to do it together. The other part as well that I like is that in the research organization, we have about half of the people come from academia. We have people that come from their postdoc, their PhD, and we have people from very different horizons. So I was sitting close to Rox yesterday, and she told me she's a material scientist. And I find it really great to see a material scientist working in plant biology. And I have a physicist working on string theory that has joined the company and is making amazing things in our capacity to analyze data. I have a chemist who has never seen a plant before, but he's working on the transformation technologies because he doesn't see a plant. He sees a membrane with lipids, and suddenly he thinks differently. And that's really cool to have that. I always dreamt to have that in my career. At Bayer, when I say, okay, let's hire a mathematician or a physicist, people were looking at me, no. We need to hire an expert in plant biology, or actually it would be good too. So I think you are the generation that can make that happen as well. Break the silos, open the boundaries, listen to people who are not educated like you, because this diversity is what we need to address these kind of challenges. And so we are working actually on tomato, on soybean, corn, and wheat at the moment. And the other thing I like as well in our setup is that we have a scientific advisory board. So people who are from different universities in Europe and in the US who are actually helping us to look at the project and are very critical. And I want them to be critical because I want to fight complacency. I don't think we are the smartest people on earth, and we don't need to have external input. So they look at our project, and in particular, these 7 guys come every quarter in Cambridge and are milling the projects, you know, to really challenge us the way we are doing things. Because given the ambition we have, we can't really, you know, be very satisfied with what we do. And when you look at the people who have brought together, who also like the story, so George Church, if you don't know him, it's this guy. Steve Jacobson is, you know, one of the Pope of epigenetics in plants. Jennifer Doudna, you could have heard this name. She's one of the co-inventors of Cas9, you know, from Berkeley. Rodolphe Barangu is less known, but was also an inventor of Cas9. Zach Lipman, I've mentioned, with the promoter bashing. Dirk Kinsey, who is in the VIB in Ghent and a really great center for plant biology, so we have collaboration with it. And Dick Flavel, who has been involved in WIT for many years and is helping us as well. So, if you want to work with us, there are some open positions. Look at the website, there will be some more coming, and connect with me here, use the opportunity. I'm here until tomorrow night. And to finish, I would like to thank you first for your attention, but I'd like also, I can't have a slide to acknowledge all the people I've been working with, because that would take 3 or 4 other slides, but I have picked a few, and, you know, different parts of my life where, you know, with the IWGSC, with the people at INRA and the Breadwheat project in France, and even at Bayer, you can have fun in big companies, you know. And now with INARI, and so I really enjoy, you know, working with my colleagues, and you see, these are the folks in Cambridge here. So this is really fun. It has been a great adventure. There are common things with this. It's working together with a group of people that you appreciate. It's trying to push the boundaries, and if people tell you it's impossible, you are the better. Let's do it because it matters. And so working together on something that matters, and then having fun is a big part of it. We're working too hard to not have fun in this job. So I encourage you also to have fun, even if it's hard during the PhD year. Okay, thank you very much.